Data scientists have questions. They are curious and want to find the explanation to different phenomena. For this reason, they use experiments to answer research questions. In this video, I will talk about the two different categories of experiments and the key ingredients to having a good experiment design. The first type of experiments are control experiments. They prove the effectiveness of a treatment by comparing a treatment group and a control group. The control group is not given the treatment or is given a placebo instead of the treatment. A difference in the response between the groups will tell us if the treatment had an effect. The most common examples of control experiments occur in the field of medicine, when a new drug has to be tested if it works or not. To have a good control experiment, it's important that the treatment group and the control group are very similar, as similar as possible. If they differ, it is possible that there are other variables that explain the difference in the response. These are called confounded factors. The best way to achieve similar groups is to have randomized control experiments. So, to randomly select who goes in the control group and who goes in the treatment group. Additionally, it's better to have double-blind experiments, which means neither the subjects nor the investigators know who belongs to which group. In control experiments, all variable measures and manipulations are controlled. This is not the case for the second kind of experiment, the observational experiments. In observational experiments, investigators do not assign subjects to treatment or control group. They just observe subjects that already belong to one of these groups. Take, for example, smokers. You cannot have two groups and tell one of the groups, hey, you should start smoking and do it for 1 year. And after that, let's observe what happened. You can't. You can only take people that have already been smoking before. One of the biggest difficulties in observational experiments is that the subjects may vary between themselves in crucial ways beside the treatment. In other words, there can be confounding variables that make them very different besides the treatment. One of the most famous statisticians, Sir R.A. Fisher, did not believe the evidence against cigarettes and suggested possible confounding variables. For example, a gene that caused both cancer and that made people want to smoke. We now know this is not true. We can make our experiments better by controlling for confounding factors. What we do is subselect smaller group of relatively homogeneous subjects. For example, instead of selecting smokers and non-smokers, we can select male non-smokers and male smokers, and the same for females. It is important to notice that observational studies can establish association between two factors, but they do not prove causation. In comparison to controlled experiments, observational experiments are weaker in terms of statistical inference, but they have the advantage that the responses are observed in the natural environment. A major goal in experiment design is to minimize or eliminate systematic errors, also called biases. There can be biases in the process of selecting subjects for the experiment or in the way the information is collected. A biased sample is a sample that is not representative of the whole population. For example, in a telephone survey, if you don't take into consideration cell phones, your sample will be biased, because nowadays many people do not have landline numbers. Not taking systematic errors into consideration can lead to false conclusions, even if the statistical procedures are correct. In the next videos on designing experiments, I will talk more about the different types of biases and the statistical tools that you can use to compare responses in experiments. For now, do not forget that a good experimental design will make an analysis much easier later on.